So NGS technologies, they helped a lot in sequencing and making the big projects which are service to the overall humanity. We pick just 5 for instance here, just to give you an idea what's going on in those finest universities of the world. So here we have the big one is ENCODE. ENCODE is encyclopedia of DNA elements, where they are interested in identifying the special transcription factors. They expect that there are around 1800 transcription factors present. So far they have identified 119, and this project has produced over 30 research papers with 50 trillion bytes of the data. You can look into its website for details. In 1000 Genome projects, actually they were interested in identifying the different variations in the human population. So they sampled over 1000 individuals coming from different parts of the world. So in total there were 14 populations. So they produced 20,000 GB of raw data, about an 5x coverage. When we talk about coverage in genome sequencing means it's a rough estimate that how many times part of the DNA has been sequenced by the sequencer. So how many times it was read. So it's like 5 times each nucleotide has been visited and read in the sequencer. They produce over 38 million SNPs and 14 million indels, and then 14,000 large deletions so far. So about 50% of them are novel, these variations, that is on average any 2 human beings they differ by about 3.6 million SNPs, over 300k indels and about 700 large deletions. So that's what they said. You can look into it in its website, the link is right there. National Heart, Lung and Blood Institute, commonly known as NHLBI, they were working on exome sequencing. So they actually provide a global leadership for research, training, education. Basically they are interested in controlling the heart diseases and lung and blood diseases. Their idea was to enhance the health of individuals so that they should not have those diseases, and then we can increase the standards of their lives. So you can look into the web link. They have yielded about 50 terabytes of data. So each sample has been having those at least 30 million reads. 50 or 76 bases paired end data. The purpose of telling you or mentioning about this data is that there is a lot of data which is available on those databases. So some of these projects are generating the data, and then what they do is they just cover one aspect in their analysis. So if we can train ourselves into data analysis, or we can further analyze and reanalyze data and find new discoveries. NHLBI also studied functional impact of rare mutations in coding sequencing using whole exome sequence data from over 2,400 individuals, and their coverage was around 111x. They sequenced exomes of over 6,500 European Americans and Afro-American individuals, and what they find here is that European Americans have a higher fraction of coding variations than Afro-Americans, so more variations in European Americans, white Americans. Another project is de novo mutations in autism. Basically there are 3 different teams coming from different parts of the world, so they were working on that. So actually they were looking into these mutations which cause autism, and what they see here is mutations present in children, interestingly, though their parents do not have those mutations. They produced over 2,000 exome sequences from 500 families. They identified the genes and their networks pertaining to autism. In human microbiome project, what they are trying to do is it's kind of metagenome endeavor. So they were trying to put the complete genomes of whatever microbial diversity is present in human body. So that was led by over 200 researchers from 80 research groups, and they were working on identifying this microbial diversity in the human body. And what they do here is they collected samples from 242 US volunteers, and they were sampling 15 tissues from the males and 18 tissue types from the females. So they also put a lot of data in the databases. So they produce 5 terabytes of genomic data, and they claim that they have covered over 5 million microbial genes.